Okay, so again, to reiterate everything we said, my name is Heba Jaamour. I'm a fifth year medical student, and inshallah, I'll be hosting your PAL session for today as your tutor. Very happy to have everyone over here right now. Whoever is attending with us right now or whoever is watching this later, I really hope you benefit from this lecture. All right. So before we dive into the topic of gastrulation, which is the topic that we will mainly be discussing in this session, I want to give you first an overview about embryology. Okay, so before we dive into the topic, we want to have a general idea of how to perceive and think of the topic in general first, okay? So when we talk about embryology, it is basically the process of moving from this to this, from just a couple of cells, from just a ball of cells, until we become a fully formed human being, subhanAllah, okay? And there are a few things regarding embryology that you have to keep in mind, because there are students that I personally know that graduate out of medical school without having a strong basis in it because they just did not grasp its concept since the very beginning of med school, all right? So the very first thing I want you to remember is that think of embryology like a story, okay? It's a story with characters, a story with the plot, a story with different events that happen. And the best way to go about this is to think of it as a story that has different events every single week. And that's actually how you study it. This is the best way to study it. You first look at the topic. For example, you see what happens in the first week of development, and then what happens in the second week of development, third week onwards, and so on. Because there are different events that happen and they're repeated in every single human being that is formed, okay? So this is the very first thing. Think of it like a story, okay? The second thing, always give a character to whatever you're studying in them, okay? That will help you remember them, all right? And whenever there is a difficult word that you are not familiar with, a very good thing to do is that you can divide it into pieces or divide it into smaller words for you to easily grasp them, okay? An example of that is, for example, let's say you don't know what embryology is. So we can divide this big word that you have no clue what it is into two smaller words. The first one is embryo, okay, which just means an unborn multicellular offspring. And then the second one is ology, which means the study of. So if you put two and two together, you would know that embryology is basically the study of an unborn multicellular offspring. And this helps you not only in this topic, but in anything you are going to study throughout medicine. If anything, I would like to give a big shout out to a project that has been run by some of my colleagues and friends actually here in uni. They basically worked on developing a booklet that has all those terms, and it's called MedSpeak, okay? This booklet has been distributed all around. So if you find it, I really think it would be a very good thing you carry around in your pocket, where basically it has different terms, okay? And suffixes and prefixes that are commonly used in medicine. This will help you a lot when you're facing new words that you're not familiar with. And I promise you it's going to happen a lot. And I promise you that it's normal, okay? It's going to be something that you learn your way through it. And you're going to be surprised at how easy it gets along the way in medicine, okay? So let's see another example of that, okay? But before that, our topic, gastrulation, is basically that the events that happen in the third week of development. But for us to keep the coherence of the story, it's important for us to review what happens in the first and second week first, and then jump into the third week, okay? Because we want to keep this chronologically organized in our heads and in our thoughts, all right? Let's start from the very beginning. You have an ova, which is the female sex cell, okay? And then you have a sperm, which is the male sex cell, right? The process of making the ova is called oogenesis. And oo over here refers to the female gamete, and genesis refers to the making or production of something. Put two and two together, and what will you get? You will get that oogenesis is basically the making or production of a female gamete. Clear? Right. What about the other side of things? Same thing. Spermato refers to a male gamete, and then genesis is the making or the production of something. So again, you put it together and it becomes spermatogenesis is the making of a sperm or a male gamete. Okay. Now, where is the setting of our story? The setting of our story happens in this very familiar picture, which is a picture of the female reproductive system, which includes the vagina, the cervix, the uterus, and the ovaries. Okay, and we don't in this lecture or in this topic, we really don't care about anything but the ovaries and fallopian tubes and uterus, mainly the ovaries. Okay, so let's look over here. Kindly everyone look over here and let's move forward to look what happens in the ovaries, okay? So it all starts when ovulation happens, which basically is the process of expulsion of the ova from over here, from the ovary to the fallopian tube, okay? Now in this process, when it happens, it starts traveling through the fallopian tube, but this is not its destination, it's just its highway. The destination is for it to get to the uterus. And if it luckily meets a sperm, then it will get fertilized and we get what we call a zygote, okay? In our scenario, this ova is lucky and it does meet a sperm. So let's look over here, exactly over here. It meets a sperm and therefore after fertilization happens, it's going to travel all the way through the fallopian tube until it gets to the uterus and implant itself. But let's zoom in at that just one more time, okay? The ova has so many different layers, and in those layers, you are going to find a sperm, or like a sperm is going to penetrate through all those layers until it gets, I don't know why this looks like this. It was supposed to look more, that was basically a sperm finds its way through, injects its genetic material, and then you get a zygote. Congratulations. طيب. Now, it's not going to stay over here. Again, it's going to make its way all the way through the fallopian tube. But it doesn't stay the way it is. It also differentiates along the way. What do we mean by the term differentiation? Because this is something that you hear a lot in your embryological topics, I'm very sure. Basically, what we mean by differentiation is the process of evolving and multiplying of the cells that are making up the zygote, okay? So how does it start? It very simply starts by the cell dividing, okay? So it goes from one cell, if it divides, then it becomes two cells, right? Two cell is called a two-cell stage. So when the zygote divides into the two cells, we call it a two-cell stage. And then it keeps on going this way. It becomes a 4-cell stage, an 8-cell stage, and so on. So it becomes a 16-cell stage, and then eventually just becomes a 32-cell stage. Now for context, other names for those stages are, for the 16-cell stage is the morula, in case you've heard that word before. And then for the 32-cell stage, it's the blastocyst. And you're going to hear this word a lot throughout this topic or this lecture, okay? Now, what happens next? Let's zoom in at this and see what's happening to the blastocyst, okay? Now, at the end of the first week, it approaches the wall of the uterus. For context, again, this is the blastocyst, which is the 32-cell stage, okay? And this is the endometrium, which is the wall of the uterus. So let me point at it right here. It's basically this over here, okay? So what's gonna happen is this blastocyst, it has two main goals, okay, that it's gonna try to achieve. The very first one is that it wants to live. So it needs nutrients to supply it with the necessary molecules and substances that can keep it alive and can keep it multiplying and growing. Okay, so it wants to implant itself into the endometrium so that it can get to the mother's circulation and get nutrients from it so that it can grow and evolve and differentiate, which gets me to the second goal of this, okay? The second one is to differentiate, to keep on differentiating and dividing and multiplying and growing, okay? First one, implant myself here. Second one, grow, differentiate, and evolve, all right? So let's see that happening, okay? This is when it implants itself into the endometrium. So the goal number 1 is done. But we're left off with goal number 2, which is basically when it wants to differentiate, okay? Now, this was supposed to be animated so that doesn't look like a bulk, but for some reason, it's not really work. I mean, I think I can make it work. Okay, yeah, okay, I can make it work over here. Basically, so we said the blastocyst implants itself into the endometrium, right? That's goal number 1. This is this blastocyst doing that, okay? Now, the second thing is that it would want to differentiate, okay? Differentiation means that it wants to divide into different layers, right? Because later on, for context, each layer is going to give rise to a different structure in the human body, okay? So we're going to go through a couple of names that are very, very essential and very important for us to go through, okay? The very first one is a blastocyst, which is something that we already covered, which is basically the 32 cell stage that we're looking at right here in this picture. So the first thing that happens is this blastocyst divides into two. The two layers are called the embryoblast in red over here, and then the second one is called the trophoblast in brown around it, okay? Now the embryoblast, another name for it is called the inner cell mass, okay? Trophoblast, another name for it is called the outer cell mass. So again, the cells that are bulked inside in red are the embryoblast, and another name for it is the inner cell mass. And the cells that are surrounding from the outside in brown is called the trophoblast, and another name for it is the outer cell mass. Now, one very important point to keep in mind is that the embryoblast eventually gives rise to the actual body, the fetus, okay? While the trophoblast gives rise to the placenta, or parts of it at least, okay? Now, it doesn't stop over here, okay? It divides even more. The embryoblast becomes the epiblast and the hypoblast, while the trophoblast becomes the syncytiotropoblast and the cytotrophoblast. A mouthful, you might be thinking, but you get used to those terms, okay? Now, this is the big picture. We're gonna go through each one of them one by one right now, okay? But I just wanted to keep this out there so that you can have the bigger picture of things and follow along as we're going through the slides, okay? Now let's talk about this again. We said trophoblast in brown is the outer cell mass and it gives rise to the placenta eventually, and the placenta provides nutrients for the embryo. طيب. On the other hand, the embryoblast in red is basically the inner cell mass, and it gives rise to the definitive structure of the embryo itself. So it gives rise to the body of the fetus. Everything in us basically comes from the embryoblast, okay? It divides furthermore. The embryoblast divides into two, which is the hypoblast and the epiblast, and the trophoblast also divides into two, which is the syncytiotropoblast and the cytotrophoblast. Now, the syncytiotropoblast and the cytotrophoblast both give rise to the placenta, okay? Or the chorionic villi, which is part of the placenta. But this is not what I'm worried about today. What I really want to discuss with you is what comes out of the epiblast, okay? The hypoblast forms extra embryonic structures, and again, I'm not really worried about that for now. What I want you to focus on is the changes that happen in the epiblast, which forms the embryo itself again, okay? This is at the end of the first week. As you can see, it says day 8, okay? Now, moving on, more stuff happen. There basically a water-filled cavity forms, or a cavity, a vacuole forms. And we call that the primitive yolk sac. طيب. Now for the primitive yolk sac, you have to keep in mind that there is no placenta formed yet. So there is no nutrition supply yet, and we still didn't get to the mother's circulation. So where does this ball of cells get the energy that it needs? It comes from the primitive yolk sac that forms. Basically, provides nutrition and gas exchange for the developing embryo. Okay? This is the end of the second week, where more differentiation happens, more multiplication of cells happens. All that happens and continues to happen until you get a cell that looks something like this. And at this stage, a gene gets activated, okay? This gene is one of the most important genes in our body. It is called the WNT gene. And this is when it gets activated, okay? But before I go on to the details of what happened in the third week, which is the main focus of our lecture right now, I would like to basically have a checkpoint and check that everything is clear. If anyone has any questions so far, please let me know right now. I'll give you guys 15 seconds exactly. Okay, so since you guys have no questions, then I have a question for you guys. Let's see who can answer it. Can someone read?
What is the 32 cell stage called? It's B, blastocyst.
Blastocyst, exactly. What about the morula? What stage was it?
The
Do
16.
you remember? The 16, exactly. And the embryoblast is
The first one.
Embryoblast again is the inner cell mass right here, the red one. Okay, the inner cell mass. Okay, that's very good. I think we can move on. Now we're gonna jump into the third week, which is when the process of gastrulation starts, which is the main topic of this lecture. But yeah, again, we said that we start by the activation of one of the most important genes in our body, and that is called WNT gene. Okay, very, 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 very important for you guys to remember the names of the genes that I have mentioned in this lecture, as it is very high yield, number one. Number two, it's always asked about in the exams. Okay, actually, like since we're talking about exams, there are two things that you guys are gonna get, or that are very commonly asked about, specifically when it comes to this lecture. The first one is the genes, as I mentioned, and the second one is what structure comes from what layer. And we're going to practice this a lot at the very end of this lecture, inshallah. Okay? So as we mentioned, gastrulation. So it starts by the activation of this important gene, which is called the WNT gene. But let's look at the end. What does this WNT gene do? And what is gastrulation? So the WNT gene starts the process of gastrulation. What is this word? Like we've been mentioning it over and over again since the beginning of this lecture. What is it exactly? It basically is the process of differentiation of those cells. The process of differentiation of those cells is called gastrulation. Okay, so the process of evolving and growth and different layers that come, we call it gastrulation. And eventually, at the very end of this process, when we're done, when gastrulation process is done, we get three different germ layers, which are the primary germ layers which give rise to everything in our body. That is the endoderm, the mesoderm, and the ectoderm. Okay, as they're stated over here. I'm not sure why I can't show my pointer, but please try to follow along on the slides. As I've mentioned, always if you find suffixes, try to dig more into it. What does this mean? How does it add to the word? Blah, blah, blah. Adds to your understanding. For example, endoderm. Endo means inside, meaning maybe it's the most inner layer. Let's see, innermost layer. On the other hand, there's ectoderm, and ecto means outside, so maybe it's the outermost layer. And then there's mesoderm, which meso means middle, okay? Which means that there is, maybe it's in the middle between the endoderm and the ectoderm, right? So it gives rise to those three different layers. And then these three different layers give rise to everything in the body, okay? To more different layers, like there are more layers that come up, but basically this gives rise to every single structure in our body. And this is the gist of it, basically. We're gonna go through the details, but to keep a general overview in your head or a general picture in your head, you get this cell, that divides until it becomes a blastocyst, and then it gives rise to different layers, and those layers give rise to different structures in our body. Now, let's see exactly how this happens, okay? And just for an analogy, okay, so that it gets easier for you to understand. Let's say we have this kid, okay? And I need your guys' interaction right now. And this kid has a goal, has a pile of Lego, okay? And it has a goal of building this castle. There are 3 different steps that this kid has to go through. What do you think those steps are? So what are the things that this kid is gonna do for him to get to his end goal, which is building this castle? Please let me know what you think.
Get the blocks first.
Okay.
Build the base.
حلو.
Then build the castle.
Okay. Okay, that's a really good one. Let's say he got the blocks, but they're a pile, mumble jumble pile, and it's like a big pile with all different colors together. So maybe
So-
one thing that makes sense is that he would
Organize it.
Exactly. He would organize it into different piles. Again, you have this big pile of Legos. So the one thing the kid would want to do so that it would be easier for him to organize and coordinate between the different colors of this castle is that he would divide white Legos on one side, black Legos on the other side, and then other colors on the other side. And then follow certain steps until he gets to that castle. Okay? Is this clear, this example? Makes sense, right? Now, the exact same thing happens in the process of gastrulation. You have this ball of cells, which is the blastocyst, and the end goal is to become a human being, a fully formed human being, okay? First, you get this ball of cells, which is like scattered, everything is all together and whatever. It separates itself into 3 different layers, which is the ectoderm, mesoderm, and endoderm, and then eventually forms into a fully formed human being. Should make so much sense, right? But let's see how this happens specifically. Now we've mentioned WNTG and it starts the process of gastrulation. Okay, how? What happens exactly? There are 4 different steps, okay? The very first one is that you get, okay, for perspective, I'm gonna keep this, basically, what is it? The upper right corner is the upper, is it right? Sorry, yeah. The upper right corner is gonna be a key for you to just keep in mind the sections that we're at. So let's say this is our blastocyst, okay? And then this blastocyst is cut cross. So we just sliced it at this purple line that just showed up, okay? So we're gonna look at it from above, okay? You guys get it? Sorry. Yeah. So we're looking at it from above. So we have this ball, we have a cross section at it, and we're looking from above, okay? So this is what you're gonna see, a compact ball of cells, cells everywhere, okay? And what's gonna happen is that when the WNT is activated, the very first step and the very first thing it does is that it separates those cells, okay? For better understanding, this is the cranial end, and cranial meaning towards the head. And then this is the caudal end, which means towards the torso, okay? So head, bottom, طيب? So the very first thing this gene does is that it separates this very compact ball of cells from each other, okay? Done. Now the very next thing that happens is that a primitive streak starts appearing. And what do we mean by the word streak? The word streak basically just means a line that forms, okay? This is what it looks like, okay? And then after that, a primitive pit is formed. And a pit from the name just means like an invagination within, or like something like, it's like a ball, okay? And then eventually at the very end, a primitive node occurs, which is just like an opening, okay? So there is an opening right now. What do you think will happen next? Very simply, the cells that were compact now are gonna fall into this pit or into this hole that formed, okay? It's gonna fall inside in a process called invagination. And from the name, I think you can start imagining how it happens. It's as if it's all going inside. It's invaginating inside because a hole formed and things started falling inside, okay? So again, this is in Word format. I linked a video in case you guys are intrigued to watch it, you can. Basically, again, to reiterate the steps, a compact ball of cells is separated by the WNT gene. And then a streak, a node, and a pit is formed, and cells fall down from it in a process called invagination. Now, all the cells that fall down, okay, cells that fall make up the mesoderm and the endoderm, whereas cells that stay above and don't fall down basically form the ectoderm. And then at the very end, the primitive streak, node, and pit degenerate. And this is the most important step in this whole process, okay? And we'll know why later on in this lecture, okay? But this is basically what happens. And we're gonna go through it again. So a primitive node appears, a primitive streak first appears, and then a primitive node, and then a primitive pit, which is the hole, and then invagination starts happening, which is some cells start falling down. Whatever cell fall down in this hole or in this pit form the mesoderm and the endoderm. Whatever stays above forms the ectoderm, okay? And then at the very end, when this is done, the primitive streak, primitive node, and primitive pit degenerate. They do not exist anymore. خلاص. They had one job and it's done and they have to degenerate, okay? Now, again, let's look at a different section now and keep an eye on the upper right corner, okay? We're looking at the sagittal one right now, which is basically a section this way, okay? We're gonna see an outer layer, which is considered the ectoderm. We're gonna see a middle layer, the mesoderm, something in the middle, okay? This is what we call the notochord, and it basically serves as the axis of a human being or of the cell, or of the ball of cells, actually. You can think of it like a vertebrae if you want, but it basically serves as the axis, the notochord, okay? And then finally, there's the endoderm, okay, which is the inner cell layer, okay? Now, another gene is activated at this point, and this gene is called the SHG gene. It stands for sonic hedgehog gene, which is a really funny name. It helps in memorizing it, okay? But those are the only two genes. This is the second gene that I mentioned in this lecture, and it's the last one as well, okay? So there are two that I want you to remember. The WNT gene, which we discussed what it does, and then the SHG gene, okay? And we're gonna see what it's gonna do right now. When this is activated, a few things happen. Basically, I want you to, I want you to look at this point of the ectoderm where it starts basically, it's as if I held it and started pinching it out, okay? After that, it forms two things. The light blue one is called the neural tube. And from the name, I think you guys can tell that it eventually forms the brain and spinal cord. And then the second one is called the neural crest cells, okay? And this basically serves to make different ganglia, nervous ganglia in the body, which helps make an organ be functional. So without the ganglia, you might have an arm, but it's not functioning. You might have a GI system, but it doesn't work, basically, okay? So those are what the neural crest cells do. Now, this is another picture just for you guys to imagine it better, which is basically me pinching out part of the ectoderm out, and then it has to fully detach, okay? When it fully detaches, it forms the neural tube and neural crest cells, all right? Now, there are a few more divisions that I want to discuss, and they're mainly in the mesoderm. The mesoderm divides into three parts, depending on the location of the mesoderm. You got the one that is closest to the notochord. We call it the paraaxial mesoderm. And again, from the word paraaxial means next to the axis, and then mesoderm is just the mesoderm, okay? Then there is the intermediate mesoderm, intermediate meaning between two things. And then lastly, there's the lateral mesoderm. Lateral mesoderm in specific divides into two. One is called the somatic mesoderm and one is called the splanchnic mesoderm. Okay, Heba, why are you giving us all those names? What is this? Too much, whatever. Okay. Yeah, I am giving this to you. So because each part of this is gonna give rise to a different structure in the human body, and you just gotta memorize them now. Those are all the layers, and then now we're gonna go through what they give, okay? So starting off with the ectoderm, which is the outermost layer, it gives rise to the skin, the nervous system, because as you saw, it gives rise to the neural tube and neural crest cells, and the spinal ganglia. So again, the neural tube gives the nervous system, and the neural crest cells give the spinal ganglia, okay? So to summarize, ectoderm, skin, nervous system, spinal ganglia, okay? Mesoderm, the middle layer, gives rise to the paraaxial mesoderm, which is the one that's closest to the notochord or axis, if you want to think about it that way, gives rise to the bones, specifically the vertebra, and the muscles and connective tissue around it. And then you got the intermediate mesoderm, which gives rise to the urogenital system. Lastly, the lateral mesoderm, I said that it divides into two. Somatic gives rise to muscles. Splanchnic gives rise to the peritoneum, which is basically just the covering above the viscera or above the internal organs, liver, stomach, and okay? Lastly, you got the endoderm, and endo means inner, okay? Endo refers to the part, gives rise to the parts of the digestive tract, respiratory tract, and several internal organs. From endo gives internal organs. Do you guys get what I mean? So this slide, I bet you're gonna get all the questions from this slide. What layer gives rise to what? The questions can be formed in, for example, what does the ectoderm give rise of? Or what layer does the skin come from? Okay, so it can come both ways, okay? Let's get to our checkpoint. If you guys have any questions, please go ahead and ask. I'll be waiting for 15 seconds on my time. Everything is clear. We're almost done. We're down to the last part of our lecture. Okay, since you guys have no questions, I have a question for you. So which germ layer gives rise to the skin? The ectoderm, the mesoderm, the endoderm, or the neural tube? Would anyone want to answer?
The ectoderm.
Exactly. Thank you very much. What layer gives rise to the neural tube?
Also the ectoderm.
What about the spinal ganglia?
Ectoderm also.
Amazing. What about maybe, let's say, the gastrointestinal system?
Endoderm.
Endoderm, exactly. One last one, and I'd be very impressed if you get this one. What gives rise to the bones of the vertebra and the connective tissue around it?
Intermediate mesoderm.
Close. Give it another guess.
Lateral mesoderm.
Yeah, it's actually the paraaxial. Because remember, it's closest to the axis, and you can think of the axis as vertebrae. Okay. But good job. You did a really, really good job. Okay, down to the last part of our lecture, which is, okay, since everything is going right, everything is seems very coordinated and organized with genes and different layers and all that, what could go wrong? We'll discuss 3 topics, and the very first one we're going to go through is the teratoma. Okay, now let's review. Remember gastrulation, we talked about the WNT gene and what it does. Let's review. You get the compact ball of cells with a cranial and a caudal end. And what the W and T gene does is four things. The very first one is that it separates this compact ball of cells into a more loose one, okay? And then primitive streak occurs, primitive node, primitive pit. And then the cells start falling down in a process of called the invagination, right? And after that happens, mesoderm and endoderm, whatever cells fall down in this pit form the mesoderm and endoderm. However, the cells that stay on top form the ectoderm. Okay. And then lastly, and the very most important step is that the primitive streak, the primitive node, and the primitive pit has to degenerate. So what if they don't degenerate? Very good question. We get what we call a teratoma. And what a teratoma is basically a germ cell tumor composed of abnormal tissue. And this is a picture of how it looks like, okay? It's a type of abnormal cell growth caused by failure of fully degenerating primitive streak, as I discussed, okay? Location of the teratoma is usually towards the caudal end, okay? Now, I would actually like to share a story with you guys. In my OB/GYN rotation, we had to rotate in different hospitals, including El-Shmesi, if you guys know it. And on my very first day, there was a surgery going on, and they asked me to scrub in and help out in the surgery. Okay, so I did, and it was a case of the doctor started discussing the case with me, and then he showed me this exact CT scan. Okay, and he's like, "Okay, what do you see in this?" And the one thing that I could pick up is that there is some kind of a growth that just doesn't fit in here. You know, it's not supposed to be here. What is this? What is it exactly? I can't tell. So later on, after the surgery that I observed, they sent this growth into the lab, and before they send it, we were able to take pictures of it, and this is what it basically looked like. So in a teratoma, because cells from all layers are in it, you get different structures, but they're not organized. You'd find teeth formed, you'd find hair follicles, you'd find blood, you'd find bone, you'd find everything. It's just not put in place because it's not supposed to be there. Do you guys get what I mean? Okay, that's for a teratoma. The second thing we want to discuss is a neural tube defect. And to review again, remember those three germ layers and how we discussed, there is a neural tube and neural crust cells, and we discussed that it's as if we're pinching out the ectoderm so that we can detach a part of it, and so that it can form the nervous system and the neural crust cells. So what happens if it does not fully detach? We get what we call a neural tube defect, and other name for it is spina bifida. Okay, so spina bifida is by definition incomplete closure or incomplete detachment of the ectoderm, resulting in what looks like this. Okay, there are different stages of it, there are different severities, but you guys don't have to worry about that detail. I just want you guys to be familiar with it. In clinical years, you are asked about this sometimes, where they go like, oh, what is the cause of spina bifida? Incomplete closure of the neural tube or incomplete detachment of the ectoderm. Lastly, teratogens, okay? Now, teratogens mainly refer to stuff that are harmful for the mother while she's pregnant. And this includes a lot of stuff, including stuff that makes sense, like smoking, alcohol, and drug intake, and others that we weren't really very aware of back in the day, especially. And just to share a story with you guys to help you remember this, there was actually this drug that was sold in the 1960s, and it was branded under the name thalidomide. Okay, thalidomide was a morning sickness drug, where basically pregnant women would take it whenever they feel dizzy. Okay, whenever they feel morning sickness, or in general to combat morning sickness. Okay. And it seemed like it was pretty effective for a good while until a huge number of those people of the children that were born to mothers that took thalidomide came with limb deformities. A whole generation was born like that because we weren't aware that thalidomide is a teratogenic medication, which is very unfortunate and is actually one of the darkest parts of of medical history in the 1960s. There are so many stories and videos that talk more about this, but this is me just teasing your thoughts a bit. You can go look into it if you are interested, and hopefully it would help you remember what a teratogen is and what it can do to a fetus or a baby. Okay? More than 10,000 babies were affected in around 46 nations. It was a huge deal. Okay, this brings me to the very end of my lecture. I'm very happy to take any questions that you guys have. And I really, really hope you guys benefited. So if there is anything, I'll be around for a bit. If there is nothing, then thank you for attending, and I hope to see you again soon in other PAL sessions. Thank you.